correction. Stop, I'll correct elimination once. You see the surface of the brain, gyrus. See you? Gyrus. And sulcus. Another gyrus. Neighboring gyrus. Here the same. Gyrus, sulcus, space, this space. And neighboring gyrus. Now, here, apical part of the gyrus, strong cover. Impossible to differentiate the layers. But if we will replace, if we will move the slide more profonder, along of the sulcus, you see white matter with blood vessels. It's envelope of the brain with blood vessels. And you see the cortex here. Here, the cells become more distinguishable, more visible cells. No, let's correct elimination. Oh, see you? Just at low magnification, we can see the cells, cell bodies. And here, cell bodies present, whereas see you this pericardia here? No, absent. So what matter? White matter. And here, gray matter. So the limit between white matter and gray matter is approximately here. Gray matter from this line up to the surface, or from the surface up to this line, approximately. More profonder, white matter. Only fibers, only axons, myelinated. Clear? Now, to differentiate the layers, we will change the order, not molecular, outer granular, no, another order. Algorithm of microscopy. No, first of all, superficial layer, outmost layer, molecular, easy to find. Surface and the layer containing few cells. Cells are rare. I'll zoom in later, but this layer is rather thick and poor in cells. It's correct? Poor in cells. Not rich, but poor in cells. Cells are rare. It's molecular layer. The first. Then move this slide and find the biggest neurons visible at lowest magnification, distinguishable at lowest magnification. Large neuron, large neuron, large neuron. So you see the neurons, not numerous, but presented here, and these largest, biggest neurons are situated in the fifth layer. It's ganglionic neurons, ganglionic layer, 5 layer, fifth. Clear? So you found the first, superficial, easy. Then you found fifth. Easy because neurons are ganglionic, large. Then you know that this less colored part of the slide, devoid of cell bodies, is white matter. So between ganglionic neuron and white matter, what layer of cortex? 6th layer, last layer, multiform. Not difficult to differentiate. So we found 3 layers already. The first one, easy because superficial. Then next layer, 5th layer, easy because cells are more largest. And then 6th layer, at nearest, below. If you will be asked, show please multiform. To show multiform, you must find firstly the largest neuron and then put the pointer below. That's all. multiform. Clear? Then, pay attention on this region, at this region. On this region. On or at. Pay attention on. Or pay attention at. Pay attention on this region. Or at. No. Grammar. English grammar. So, you see numerous cells. And you see that some of them are pyramidal in shape. It's at low magnification. These cells, small, 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 are pyramidal too, but at high magnification. So, this layer containing more cells than another, cells, layer more rich in cells. This layer, this one, this layer, it's pyramidal layer. Third, pyramidal layer. You found the first, the third, the fifth, and the sixth. So, second and first. To show external granular, put the pointer here between molecular and the pyramidal in this region. Put the pointer and see. It's outer granular. If you want to show inner granular, put the pointer between largest neuron and pyramidal neurons here and say it's inner granular layer. Is it clear? In what way we can differentiate the layers in the section, in the slide? Understand? Good. Now let's zoom in. Let's zoom in. Zoom in. No. Molecular. Single cells, rare cells present, but numerous. Let's zoom in more. Molecular layer from the surface up to this layer. Single neurons and numerous fibers present in this layer. This layer is rather thick. Next. Rare oval or polygonal neurons are visible, but the pyramidal neurons appear immediately. So its pyramidal layer begins. But this small amount of cells will correspond to outer granular. But pyramidal begins immediately. It means that outer granular is poorly developed. Here. Yes? Pyramidal neurons, see you. Neurons are pyramidal in shape, pyramidal, then pyramidal, and at last, where? What? No. This one. Oh, this one. Oh, see you. It's ganglionic, ganglionic. So above ganglionic, here in this region, there are few cells that form inner granule. But in this section, in this slide, granular layers are poorly developed. Well, to say honestly, can you reveal the granular layers? Can you? As me, I cannot. To say honestly, I cannot reveal, I cannot differentiate granular layer in this section because these layers are poorly developed in this section. So, from what region of the brain the specimen was obtained to slide preparation? Motor region. It's motor region. What type of cortex see you? Morphological type. A granular type, typical for motor region. Clear? When you will study the slides, try to differentiate, try to precise what type, what morphological type have you in your section. But top secret, all slides that we use to demonstrate the brain, to demonstrate to our dear students the brain structure, all slides was prepared from specimen obtained from the central gyrus of the temporal region. So it's motor region of the brain. So all slides, all cortex will be agranule. All slides, but keep silence. Top secret. Как они? Шпион, как это? Spine. Spine surround us. Attention, please. Spines. That's why. Keep in your mind. And when you will be asked, during the exam, for example, you will receive cortex, cerebral cortex, this slide. You will recognize, you know histology, you recognize this slide, you answer, oh, I have cerebral cortex, section of the cerebral cortex. And the angry teacher asks you, what morphological type of the cortex see you in the section? Morphological type. What morphological? Wait. I see that granular layers, outer inner granular layers are poorly developed, invisible, indistinguishable in this section. It means that it's a granular type. Typical form motor region of our brain. Your answer is excellent. Spine. Spines in another groups. Spines. Well, so it's the first slide. You will examine and compare with the microphotographs in your atlas. Unfortunately, we have only 5 slides for cortex, cerebral cortex, and 5 for cerebellar cortex. I'll explain briefly next slide, but you can revise spinal cord and ganglion if you want slides present here. So during the time that we have 1 hour, else you will examine the slides. But now, cerebellum, cerebellar cortex. You see that surface, the cerebellum is turned down. Section looks down. Surface of the cerebellum is here. Understand? So you see gyrus, and sulcus between. So gyri present in cerebellum too. Surface is irregular. You have the picture at low magnification. Shoot it at low magnification. And you see that impregnación on the top of gyrus, impregnación is intensive, and the layers are not distinguishable, but more profunda, the layers in the cortex is visible. And you see sulcus, it means that it's surface, yeah? Then gray matter and white matter. Here neurons absent, cell bodies, only fibers. So white matter and gray matter. But in the cerebellum, gray matter contains only 3 layers, not 6, but 3, because evolutionary it's more elder part of the brain. Clear? 3 layers: molecular, ganglionic. Why ganglion? Neuron cell, large, big, visible at low magnification, yeah? And the last, 3rd layer is granular, or layer of the granular cells. Only 3 layers. If you zoom in, you will see that ganglionic layer contains cell bodies of the ganglionic neurons. Sometimes we name these neurons Purkinje cells, because Purkinje described as anatomist. Purkinje cells, piriformic cells, piriform cells, piriform, shape of pericardium, or ganglionic neuron. Pericardia form ganglionic layer. This part of the cortex is molecular layer, and molecular layer of the cerebellar cortex. Wait, I'll correct the elimination. Oh. Что такое? Не нравится мне. No, tissue is slightly detached, destroyed during fixation, yeah? But it's pericardium of Purkinje cell, and dendrites. Dendrites penetrate into molecular layer. Here you see the surface of the gyrus, so it's molecular layer. Numerous dendrites of Purkinje cells, and another cells present here. What cells? Stellate cells, basket cells, present in this layer. Granular layer, numerous granular cells, and Golgi cells form this layer. Then white matter. So cellular composition and number of layers in the cerebellum is more simple compared to cerebral cortex. Only 3 layers here. So you must find these layers, indicate the cells present in this layer. If you will not find some structures, I'm here, ask me. Understand? And finally, I'll show you a scheme. I'll show you a scheme that demonstrates interneuronal interactions between neurons in the cerebellar cortex. Molecular layer, ganglionic layer, granular layer, white matter. Red fibers are afferent, bring the impulses in the cerebellum. Black, axons of Purkinje, is efferent, out. The rest neurons are associated. But what is the role of the cerebellum? Main function of cerebellum. This part of the brain, what is the function of cerebellum? You have you cerebellum? Me too. What for? No? Sorry?
Control the
control. I will say not only control, but correlate, regulate, control, regulate. Equilibrium, control, regulate the activity of muscles, position of our body, vertical. And we feel our body in the gravity. We don't feel gravity, but gravity influence on our body, yeah? So vertical position. When I turn my head, or when I turn with my leg, head down, I feel position changes. When you use the bus or metro, and the bus stopped, suddenly stopped, what are you doing? You run. You try to guess, to maintain position, maintain equilibrium of our body. Yeah. In what way? Who regulates these activities? So impulses from the eyes, from the organ of equilibrium. There are specific sense organ of equilibrium in the inner ear, from the eyes, from the muscles, proprioceptive, afferent impulsation. All these afferent impulses from different receptors enter in the cerebellum, in the cerebellar cortex, with help with two types of axons. What fibers bring impulses into the cortex? What? Afferent, right? But how we name these afferent fibers? Look, one of them signs with the dendrites of Purkinje directly, red. See you? Branch along of dendrites and synapse with dendrites of Purkinje. One fiber, one cell. What fibers? Climbing fibers. And there are mossy synapse with the granular cells in the granular layer. So afferent impulses are transported into the cortex of cerebellum. What neurons transport impulses out the cortex from the cerebellum? Ganglionic cells, Purkinje cells. Their axons enter in the white matter. So efferent impulses from cerebellum are transported, are conducted along with these axons. So there are afferent, there are efferent chains. And between neurons that bind, interneurons that bind, afferent, efferent. Is that clear? So, look, mossy fiber, then granular cell, axon into molecular layer, branch and synapse with dendrites, but dendrites of different, not with one, but different Purkinje cells can receive impulses from one granules, because axon is long and branch. The same basket cells. Axon binds the Purkinje cells. Stellate cells, interneuron. Golgi cell, interneuron. But the problem is, what neurotransmitters are released by these interneurons? If neurotransmitter provoke depolarization of Purkinje, of membrane of Purkinje cell will be excited. If neurotransmitter provoke hyperpolarization, Purkinje cell will be inhibited. So it depends of neurotransmitters secreted, released by these interneurons. And in the cerebellum, granular cells received impulses from mossy fibers, granular cells, mossy fibers and granular cells. And climbing fibers are excitatory. Nervous impulses transported through these neurons, through these fibers, excite the Purkinje cells. The rest neurons present in the cerebellar cortex are inhibitory neurons, produce another neurotransmitters. And now imagine climbing fiber and basket cell. Climbing fiber is excitatory, basket is inhibitory. And the question is, what impulse will be the first? If impulse from climbing fiber will be the first, the cell will be excited. But if basket cell send impulse, and this impulse promote inhibition of Purkinje, climbing fiber cannot excite because neuron is inhibited already, and it's necessary to spend the time to repolarization will take place. Is that clear or no? So in our cerebellum there are group of Purkinje cells scattered around of the cortex. One group is excited at the same time. At this moment, these Purkinje cells are excited. This group is inhibited. Axons from these neurons go to the motor neurons in the spinal cord. So one motor neurons will receive impulses and will be excited. Another group of motor neurons will be inactive. Different groups of motor neurons innervate different groups of muscles. Flexor, extensor. Flexor, extensor. When I walk, alternative contraction, contract, relax, contract, relax, contract, relax. So who regulate, who control? Different group of neurons in cerebellum. Clear? So this picture, this scheme may be, I'm not sure, but maybe during exam you can receive. During exam you will receive microphotographs, yes, during control. Control problems, schemes. Maybe this scheme will be present. That's why. Shoot and try to revise once more. And if you will have the questions, I am ready to help. Clear? Now,